Hi, welcome back. We're heading near the end of the term, and today we're going to talk about plant hormones and some tissue culture stuff. So let's get right into it. Um, some of the key concepts I hope to cover is, how plant hormones influence growth and development, and some of their applications. We're not going to cover every single type of hormone that's out there. There's been a lot of research in recent years and new classes, but all the ones that are important for horticulture and practical applications. Um, Hormones are responsible for creating what we call predictable patterns of development. It's a fascinating area of research because if you take a cell and you divide it into two cells, and then two cells into four cells, and then four cells into eight cells, if nothing really happened, they would just keep forming a big ball, like a big basketball, and then eventually just a big mass. At some point, the cells have to be able to communicate to each other that, okay, look, you're going to become a leaf, you're going to become a root, you're going to become a stem, and all those different structures that we're familiar with, those come about because of plant hormones in different concentrations in different locations, and they impact each other to elicit these outcomes, and we call that predictable patterns of development. Um, Plant hormones are also used to measure changes in the environment so that plants can time their different forms of growth with the right season. Um, and so we'll look at how those things all play into with tissue culture as well. So there's a list: auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Um, Auxins, we'll start with them, are typically promote cell elongation. They have an important role to play in what we call phototropisms and gravitropism. So tropism means it's a movement. So photo means movement with respect to light, and gravitropism means movement with respect to gravity. So auxins are typically found above the soil line in the shoots, for the most part, in meristems, and they travel down through the plant for their eliciting their influence on growth. And of course, indole acetic acid is one of the most common. It's not the most common in terms of synthetic rooting compounds, but it is definitely the most common one out. This is just an early experiment showing where a coleoptile is cut off and then it's placed onto a gel and allows, with some time, auxin diffuses out of the growing tip into the gel. And then they take the gel and they cut a small piece and they lay it on half of the coleoptile. And of course, what they're trying to understand is, okay, so there must be something in the tip, right? And that is in the gel now, and it diffuses down one side of the coleoptile, but not the other because the gel is on only one side, causing it to bend. And that's how basically they started to understand what auxins did and how they impacted cell growth. Looking at tropisms, as far as gravity is concerned, in this picture here, we're looking at a microscope image of a root tip. And zooming in on the cells, there's little granules. Um, we call them statoliths, and they're basically starch granules. And they're like a little weight, and they tumble to the bottom of the cell. And in this case, the root, let's say, is facing directly up and down, so it's vertical. So the statoliths are right at the bottom of the cell. So the root is going to continue to grow straight down. Okay, it's not sensing any angular position of gravity. If we were to take this root and turn it on its side, the statoliths would all tumble to the side of each cell. So they'd all be on this side of the cell. And what ends up happening is because they're on the side of the cell, they inhibit the growth of auxin on that side, and then this side continues to grow, which drives the root down. So the root is always being driven straight down with respect to gravity. If you look in your PowerPoint, there's some video here that will help explain this if you want. NASA does a lot of research on this because trying to grow plants in space is very challenging because gravity is obviously really not present. So these statoliths are really confused and they're floating all over the place, and it's hard for the plants to grow normally. So trying to find a way to get the statoliths to respond to something, you can put the plant in sort of a spinning chamber so it simulates gravity. And that's typically what they do in the space station so that the plant grows normally. Then with phototropism, like in this plant here, the plant grows towards light. Now, my next slide is going to help with this. In this case, what happens is sunlight hits the side of the stem and inhibits auxin on this side, which means that auxin is normal on the backside, which causes it to grow and drives the stem towards the light. So this ivy plant is growing towards a light because the auxin is inhibited on this side, but not on the dark side. There's another picture depicting that. We'll just keep going here. So auxins in horticulture are applied to crops to prevent leaves from falling off at low doses, to prevent fruit from falling off. They're kind of related in the process. It can also help in promoting flowering and fruiting. And down here, it's a very common rooting compound. So we dip cuttings in auxin and it helps stimulate roots. And then over here, we see that it's also used to control weeds. 2,4-D is a synthetic auxin that when you apply it to weeds, particularly dicot weeds, it causes them to accelerate their growth beyond their ability to make the energy to support it. So if you spray a dandelion, it starts to really go tall and curls around on itself, and eventually it outstrips its own energy. The photosynthesis can't keep up with the pedal to the metal growth that high dose of 2,4-D is giving the dandelion. So 2,4-D doesn't actually kill the dandelions through toxicity. It kills the dandelions by shifting their growth patterns to basically full throttle where they can't keep up and they end up starving. Why is it such a bad thing? Because when they manufacture 2,4-D, it's pretty much impossible to prevent dioxins from coming into the chemical. And dioxins are potent teratogens, which means they cause birth defects, and carcinogen. So for that reason, 2,4-D has been banned for residential use, and landscapers have to have a license, a pesticide license, to apply it safely. It doesn't mean that it's a good idea for you to use it on your lawn, if you ask me, because if you like to stand or lay on your lawn, or if you have pets, you don't really want them walking through that dioxin in the grass. That's not good. Yes, it was used as a herbicide during the Vietnam War, and that's why all the veterans started getting sick, was because of the high dioxin content in the Agent Orange, which is a mixture of 2,4-D and another synthetic herbicide. So one of the sadder chapters of horticulture there. Now, auxin is what we call, it plays a key role in what's called apical dominance. So in this picture, we have axillary buds that are sitting here dormant, right? And they're not growing because auxin is being sent down from the apical meristem and suppressing them. So that's an important role that auxin plays. If we cut that top off, so here we've severed the apical meristem, the lateral branches actually grow. And in horticulture, we rely on this, right? We prune plants to encourage branching so that it's more full. The classic example around Christmas time is Christmas trees. So Christmas trees, if you look at say a spruce or a balsam that's growing in the wild versus one that you buy at IKEA for your Christmas tree, the one that you buy at the store is way denser with far more branching. And that's because the apical meristems on each branch are constantly trimmed. They're hacked, which breaks dominance on the branches, causing them to branch out and become more full. So not only do we have apical dominance at the top, but the branches also display apical dominance. The other thing that's interesting about conifers is that the main leader exhibits the strongest apical dominance, and the further away you get from the top, the less inhibition there is, which gives you that classic conical shape in a conifer. Here's a picture of someone using a backpack gas-powered Christmas tree pruner. So they would just walk around and maintain trimming the tips, encouraging lateral branching so the tree becomes more and more full. That doesn't look like much of a Christmas tree to me. That looks more like a cedar, but whatever. Moving on to gibberellic acid, that's the second hormone on our list. Gibberellic acid is used for a lot of really interesting outcomes. In the case of blueberries, it encourages fruit set. So commercial growers will apply gibberellic acid to enhance the berry count on their plants. Gibberellin principally is something that's used to promote stem lengthening, stretch. It's used to break dormancy of seeds and buds. Plays an important role in flowering and fruit set, like with the blueberries. And it's also in apical meristems. It's in buds. It's also in roots and leaves. It's basically all over the plant. It's kind of an interesting plant hormone. And it was first discovered in rice, and it was the result of a fungal infection that caused rice to grow kind of tall and lanky, like these long stranded rice. They were given the nickname foolish rice. That's kind of an interesting process. So the name of the fungus that was infecting was called Gibberella fujikurooi, which they discovered stimulated cell elongation and division. So things got really tall, not just stretched, but grew. So it was first extracted from this fungus to make it. Now it's made synthetically. Um, So gibberellin, one of the key things to remember is unlike auxin, which causes cell elongation, gibberellic acid causes cell division and elongation. So you get way taller stretch in plants sprayed with it. It's not just about stretch like phototropism. We're talking like serious stretch. Um, a lot of times dwarf varieties, like you'll see an advertisement for a seed at Stokes or whatever that says dwarf variety. So usually they're deficient in gibberellic acid so that they are compact. And you can spray them with gibberellic acid and they will restore their growth pattern. Classic example is if you spray cabbage. So in this picture, you can see that the stem of the cabbage is quite compressed. So here's your apical meristem. And all the leaves are rolled up into each other, right? It's absolutely compressed. And so this really cool old photograph that I got off the internet from the Life magazine, you'll see I zoomed in here. They take a cabbage and they spray it with gibberellic acid and it stretches to like 3 meters tall or more because this stem is no longer devoid of gibberellic acid, so it stretches out and the leaves have ample room to have proper internodes. So it's kind of an extreme example of what gibberellic acid will do. So gibberellins are involved in overcoming dormancy in seeds. They move throughout the plant in all directions, produced in all tissues pretty much. If we look at seed dormancy, the way it works is that if you imbibe a seed, so you soak it in water, it triggers the release of gibberellic acid from the embryo. So this is my icon. This is the embryo inside the seed, and this is the cotyledon, let's say, and it's surrounded by the seed coat and the seed wall. The gibberellic acid moves out of the embryo, right, through the cotyledon and gets to the seed coat, and there it stimulates the production of something called alpha amylase. Alpha amylase is a really common enzyme that breaks down starch to make sugar. It's in your spit. When you put food in your mouth, your saliva begins the digestive process because amylase is in your saliva. Anyways, that starch that's in the cotyledon begins to release sucrose from the starch. And that sucrose moves into the embryo and begins to drive growth. And you end up with germination. So by applying gibberellic acid externally to seeds, we can actually accelerate or break dormancy that's waiting for specific requirements. So gibberellic acid is released from the embryo. Amylase is produced, which goes to the seed coat. That produces or breaks down starch in the endosperm and the cotyledon, and then the sugars are used to drive the growth of the embryo. So typically it's used to break what we call stratification. So seeds, typically seeds from perennials that are expected to sit through a winter. So you have your perennial flowers all summer, produce the seed falls in the ground. It would be a bad idea for the seed to germinate typically right before winter because the plant's too tender, it might freeze. So the seed is programmed to lay in the ground and it tracks the cold winter. And it knows that when winter is over, then it can germinate. And so we simulate that by putting seed in freezers. And that's what this picture is trying to show here. It's got some lavender seed in the freezer, for example. And sometimes you don't have enough time to properly stratify the seeds. And so what we do is we spray the seeds with gibberellic acid, just like on the previous slide. And that breaks the dormancy. Ideally, you put the seed in the freezer and you give it a proper stratification. But if you have to, you have gibberellic acid as a fallback. In terms of promoting flowering, the classic example is azaleas. This is an ornamental azalea, so it's been grown to sell in a pot. And all the flowers are happening at once, where typically the azalea flowering is spread over a longer period of time. But when people want you to buy it, you have to have all these flowers to attract your attention. So by spraying the azaleas with gibberellic acid, we break the dormancy in all the buds and they flower at the same time. And you have this real fantastic show. Another common application for gibberellins is in the produce industry. What you have here is on the right is seedless grapes that from California that have been sprayed with gibberellic acid versus control ones on the left. And so most of the grapes you buy at the grocery store have been sprayed with gibberellic acid. And the reason being that the farmer gets more fruit off their field. It's not as tasty because basically you take this grape, it's got the same amount of sugar as one of these little grapes. But if you're selling them in a fixed package or by a unit, this cluster is going to get more money because it's full of more water, right? So yeah, that's how the system works. So if you're complaining about the flavor of grapes, it's because the sugars have been diluted. Now, wine grapes, for example, we don't want water. We want them concentrated. So you'd never spray those grapes with gibberellic acid. But table grapes, yeah, they're sprayed. So summarize auxins, tropisms, rooting, apical dominance. And for gibberellins, we have cellular elongation in combination with division, right? They also have a role to play in germination and flowering. So let's look at cytokinins. So cytokinins promote cell division, right? So auxins are cell elongation, gibberellins are cell elongation and division, and cytokinin is cell division. Cytokinins are found all over tissues, basically wherever there's lots of cell division happening. So embryos, in fruits, meristems, that kind of thing. Typically, I would say the cytokinins for the most part are produced in the roots, like larger quantities. And it's most dominant, for example, in trees as they go through the season. You end up with a lot of cytokinins in the roots throughout the winter. And then in the spring, the sap starts to flow and it drives up into the plant. That moves up the plant and it helps break the dormancy in the buds, right? So if you see your fruit trees, for example, they know when spring is happening, they begin to flow their sap, and that sap brings the cytokinin up to the buds, and then that breaks the dormancy in the buds. Cytokinins are also used to delay senescence. So senescence is a fancy term for death. In leaves, what happens with death is they lose their chlorophyll, they become yellow, and then they become necrotic, or they start to die, and then they fall off. And so what we have in this picture is a couple of tobacco plants. And on the one, the lower leaves have been sprayed with cytokinin, and the other they have not. And a lot of times these plants, they sort of have programmed cell death so that the lower leaves are meant to dry up and fall off at a certain point. But sometimes in ornamental crops, we don't want the leaves to fall off, so we can spray with cytokinin to prevent that leaf drop from happening. Cytokinins are also really important for something called morphogenesis. So morphogenesis is like the shaping of a plant, sort of like how I started the lesson talking about plant and hormones and understanding what needs to go where in space and time. Um, Cytokinins are really important in combination with auxin, and the ratio of the two is really important. We'll talk more about that as we go into the lesson and talk about tissue culture. But on this example here, the classic example is a bonsai. So bonsais, typically you have to trim the roots and then you have them in very confining pots. And that's designed to suppress the growth of the root tips. And by doing so, we are lowering the cytokinin content in the phloem. And as a result, with this imbalance between auxin and cytokinin, the bonsai doesn't get the right signals and the plant stays really stunted and the cells don't expand and divide very much. And so you end up with this really small dwarf version of a tree. So we're looking at cytokinins. They promote cell division. They enhance lateral development. We looked at fruit trees. So in the spring, when they flow up in the branches, they trigger the development of buds. They delay senescence, the aging and dying of old leaves and tissues. And in tissue culture, they're used for morphogenesis. We'll touch more on that after we're done talking about the hormones. So I'm going to skip ahead. So with auxins, they're responsible for cell elongation. We talked about the different tropisms. They're used for rooting and apical dominance. Gibberellins cause extensive cell elongation in combination with cell division, critical, for sure, remember that. They help with germination and triggering of breaking dormancy. They also stimulate flowering, like in the azalea. And cytokinins stimulate cell growth and bud development and also delay senescence of lower leaves. So let's look at ethylene. We're not a chemistry course, but I'm just showing you this because ethylene is actually a very small, volatile chemical. It evaporates into the air very easily and spreads through the air. So plants are able to detect it by just being in the presence of the gas. It's very small and evaporates easily. This is a plant that has had ethylene damage. The leaves are curled, the buds are damaged. It's had a really negative impact on the plant. Now, ethylene was first discovered in the 1800s. Believe it or not, we've had ornamental plants for a very long time. And many, many years ago, they used to have gas-powered lanterns in the streets so that people could walk in the evening and not stumble around in the dark. And they would have plants near these lanterns. And it was noticed that plants that grew right near the lanterns grew unusually. They had short, thick stems. The leaves would fall off. They didn't look healthy. And it was eventually someone discovered that this was because of a gas that was a byproduct from burning the combustion gases in the lanterns. So one of the byproducts from burning that gas was ethylene gas, and it was a powerful growth hormone. So one of the classic experiments is you take an apple, which produces its own ethylene, lots of it, and you put it under a beaker with a branch of leaves. In this case, it's holly. In the control, the plant's sitting in the water and it's getting light. It's fine. It's happy. But in the beaker where the apple was present, the ethylene is causing the leaves to fall off. So we call that leaf abscission in combination with senescing, but it's primarily leaf abscission. So ethylene causes leaves to fall off. It also causes fruit to ripen. I mentioned the apple. Got a couple other examples coming. And then on top of that, it also causes the degradation of chlorophyll. So we call that degreening. So in oranges, lemons, and grapefruits, that's a common industrial practice to make them look more ripe when they come to the grocery store. They're gassed with ethylene to make the rind more orange or yellow in the case of the lemons and grapefruits. Because having lots of chlorophyll in the orange rind makes you think it's not ripe. But most oranges actually still have chlorophyll in the rind when they're ripe. So ethylene triggers leaf drop. And as far as in horticulture, the classic example is hydrangeas. We stick cuttings and then we grow hydrangeas outside in the summer till they're a nice bushy plant. And then we expose them to ethylene, which causes all the leaves to drop. And then we end up with sort of like dormant sticks in pots that we put into a cooler, and we treat them with a cold temperature for a period of time. And then we stick them in the greenhouse and we force them for spring. And we end up with hydrangeas that are growing ahead of time with nice flowers. And ethylene is an important part of that process because we need to drop the leaves off before they go in the cooler, so we can control when the leaves come off and not have a lot of rot. As far as fruit ripening, you may have heard the phrase one bad apple spoils the bunch, right? And the idea is that once a fruit begins to ripen, it starts to produce its own ethylene, and you get a chain reaction. So having one overripe apple beside other fruit or beside other apples will accelerate their ripening. The classic example, of course, is bananas, right? So apples and oranges produce a lot of ethylene, so you should never store your bananas beside apples and oranges because their shelf life will just be diminished really, really badly. Um, Bananas are harvested green, and then they're put into a shipping container, and then they put an ethylene pill inside, or what we call an ethylene bomb, which exposes the bananas to ethylene so that they begin to ripen. And by the time they arrive at your grocery store, they're well on their way in the cycle of ripening, and they're producing their own ethylene, which is then accelerating the ripening of anything that didn't get exposed to ethylene. I already mentioned the degreening of oranges. So this is what oranges typically look like when they're ripe. Not nice and bright orange, but the pigments, the accessory pigments that are not chlorophyll, like carotenoids and anthocyanins, they're not degraded by ethylene, so they're left behind, which gives you that nice orange color. Same thing with leaves in the fall. The trees produce a lot of ethylene that causes the degradation of the chlorophyll and leaves behind accessory pigments in the leaves, which gives us the nice yellows and oranges and reds. What's important to note is that the plant doesn't waste it. The chlorophyll that's dismantled, all the nutrients that are in there are taken through the petiole and stored in the stem for the next year, so it's not wasted. And then, of course, the leaves that fall on the ground, they compost, and the nutrients that are not able to recycle directly are recycled through the soil. Ethylene is also responsible for something called epinasty, which is when leaves curl in response to stress. Tomato plants will do this, for example, when they don't have enough oxygen in the root. So you get a lot of rain if it's in your garden, the leaves will curl. Or if you're growing tomatoes hydroponically and they're just too wet all the time, the leaves will curl, as an example. So epinasty is sort of a stress response. A more obscure application of ethylene is the treatment of seeds to make sure that they're female. So cucumbers have male and female plants, and commercial growers want to grow seedless cucumbers. So cucumbers don't have to be fertilized, they just need to be female. And it was discovered that if you treat cucumber seeds with ethylene, the seeds will almost all be female. And so then the farmer knows that if they're going to plant 50,000 cucumber plants, that they're all going to be female, because the male ones aren't going to make cucumbers. They won't be of much use to you. So summarizing ethylene, it's a gas, right? Rapidly diffuses through the air and through the plant tissues. If it's produced by one plant, it affects nearby plants. It's involved in fruit brightening, encourages senescence and abscission. That's where fruit falls off. It also inhibits flowering in most species, but will promote it in a few, like pineapples and mangoes. It's also responsible for epinasty, the downward curvature of stressed plants. And it's also useful in determining the gender of certain plant species like cucumbers. Just to touch on abscission, we'll move on because in addition to ethylene, abscisic acid plays an important role. Both as a growth inhibitor, but also in dormancy and in abscission. So let's look at this. So as a whole, think of abscisic acid, otherwise known as ABA, as an inhibitor, not a growth promoter. So we're not looking at cell elongation or cell division or promotion of lateral growth or anything like that. It's an inhibitor. It's to prevent. Okay? So it prevents buds from germinating in the spring. And for example, in the case of buds in the spring, the rain washes some of that abscisic off, allowing them to emerge and begin to grow. So the tree, in a sense, knows that the rain has happened. Very similar with seeds. A lot of seeds, when we talked about this, you have to imbibe the seeds. You have to soak them first for a little while. And part of that is to soak off abscisic acid because it's in the seed coat. And as long as the abscisic acid is present, the seed's germination is inhibited. And again, this is an adaptation by the plant so that it knows that spring rains have come, or if it's a tropical country, that the monsoon season has passed. And that way the plant can measure the amount of rain and it knows that, okay, it's rained enough, now it's safe to germinate and I won't get drowning in water. The other thing that abscisic acid does is cause stomatal closure. So your stomates in your leaves, when there's any kind of water stress or heat stress, abscisic acid will close the stomates to stop water from evaporating out of the plant. It's not a great thing to happen, but if the plant's getting to critical levels where cellular damage can happen because of wilting, abscisic acid can reduce the damage by closing the stomata earlier. Looking at abscission or leaf or fruit drop, the way this works is that as the days get shorter, the plant will change its ratio of auxin to ethylene. And once the plant is committed to the leaves, to its sort of comprehension that fall has finally come, it releases abscisic acid, which will then basically degrade the way the leaf is glued on. So if you look at a leaf where the petiole attaches, and this is a close-up here, where the petiole attaches to the stem, there's a gap. And inside that gap, there are pectins or types of sugary glues that hold the leaf on. And you can snap it yourself. You can break it if you want. But in the fall, once abscisic acid is produced, that starts to degrade the pectin, and the leaves will start to fall off when there's wind or just by gravity. So that's abscission, right? Same thing as a fruit ripens. When the plant decides that the fruit is sufficiently ripe, it produces abscisic acid. The abscisic acid breaks down the pectin where the fruit is attached, and then the fruit falls on the ground. So we've covered auxins, gibberellins, cytokinins, ethylene, and abscisic acid, which positions us now to talk about tissue culture. So what is tissue culture? It is a means for reproducing plants without going through fertilization. So we call that asexual reproduction. And it's the key thing here, remember this for your exam, totipotency. The key thing here is that plants have a really unique ability in that you can take a single cell of a plant and you can produce an entirely new plant from just one cell. And that's where we get the term totipotency, the ability of a single plant cell to give rise to a whole plant. And that's really what tissue culture is all about. Now, we don't always use a single cell. We usually use pieces of plants, as you've done in your lab. This is a picture of a research lab. This is someone working in a commercial tissue culture hood where you're trying to keep bacteria out and plants tissue sterile. What are some of the uses for tissue culture? A lot of specialty plants are propagated through tissue culture because it's just easier. It's too difficult to go through traditional seed and germination cycles. Orchids are the classic example. Tissue culture is used for genetic engineering. If you want to introduce a new gene into a plant, you have to rescue the plant after the process and regenerate it through tissue culture. Quite a few plants are used in tissue culture to produce drugs that the pharmaceutical industry uses and sells to people. Oftentimes they grow sort of tissues from plants in a bioreactor and they circulate in media. And while they're growing, they produce these chemicals which go into the liquid, which then they can extract and sell as medicine. They also are used to produce enzymes for the food industry. Vaccines are produced using tissue culture and plants. There's links here to Medicagos based in Montreal. They're also algae bioreactors, which use a lot of the similar ingredients to tissue culture. It's a fine line between what's tissue culture and what's not, but I'm giving you some of the broader applications of the technologies that we essentially use tissue culture in liquid form or plating on media. There's lots of different strategies. We could go on forever, but this is not meant to be exhaustive. From a day-to-day practical perspective, I don't know about you guys, but I love bananas. All bananas are grown through tissue culture. All banana plantations. So basically, bananas are all clones. And you've probably heard the story of Cavendish, the original banana variety that we don't have anymore. We're on another variety, but what ends up happening is when everything's cloned, there's no genetic diversity. And so when a fungal infection breaks out, it wipes out the entire industry. And we're on our way out with the current variety. So a new variety has to be made to replace that so that we can continue to eat bananas. And honestly, I already confessed that I like bananas, but truly, bananas are probably one of the worst things ecologically for the planet because the farming that is done to grow bananas is so intensive. The pesticide levels are so intense. The nutrient removal from the soil is so intense. And we're talking depleted tropical soils that once the plantation is done after a few years, nothing grows on that field anymore. So it's really, they really are a disaster. Anyways, why tissue culture? So we can produce disease-free plants. So that's called meristem culture. We can also rapidly produce a lot of plants in a very short time, which can be really useful for certain varieties that are difficult to propagate. It's also obviously very good for difficult to propagate, like orchids. We get with tissue culture plants, the plants that come out the other side typically have more consistent growth and a more even canopy. We already talked about hydrangeas earlier in the lesson. So for example, hydrangeas are a woody cutting, and it's very difficult to get really consistent growth with woody cuttings. I mean, it can be done, but it's difficult. And some people actually grow hydrangeas through tissue culture, end up with laser level, super uniform crops. So sometimes even get more flowers or more yield if it's produce. There's a lot we don't really understand why that's the case, but that does often happen. You can have year-round production for crops that might otherwise need a cycle or a break. And again, I mentioned before, the tissue culture can also be used to rescue genetically engineered plants. Um, Limitations, it's expensive, requires a lot of skill. Contamination, as I think you all know, is a major concern because we have a nutrient-rich medium there, and all it takes is the tiniest spore or a single bacterium to get in there, and it will just take over. One of the real challenges with tissue culture is acclimating the plants once they come out of tissue culture into soil. That's a very difficult transition and takes quite a bit of skill to do successfully. And lastly, depending on how well the protocol is done, if your plant hormone levels are just a bit too high, they trigger genetic variability, and you end up with plants that are not true clones. And interestingly, this is how a lot of annuals are bred by causing what we call mutagenesis through tissue culture or chemical stimulation to mutate the varieties to create a new petunia or whatever, lantana. That's not genetic engineering per se because it's sort of happening naturally, but tissue culture plays a role in that. Micropropagation is another term for tissue culture. Um, it is an in vitro process, which means it's in glass. In vivo means in organism. So we're growing in a plastic container or a glass jar under aseptic conditions. Um, Three different kinds of tissue culture starting points. One is organ. So organ would be like you take a shoot, like a potato, and then you stick that into basically a tissue culture media that has mostly auxin that triggers rooting. It's pretty simple. It's not like a multi-step process. It's almost not really tissue culture because you're really just rooting a cutting. Um, but then there's seed where you throw a seed onto the media and then germinate it. That works really well for plants that it's very difficult to germinate the seed reliably. Or if you're preparing a plant for genetic engineering and it needs to be sterile, then you can grow the plant from seed in tissue culture. The most common is what we call callus culture from vegetative tissue. And I should mention in the Oregon one, meristem culture, as it says there. Remember, the meristem is the growing tip of the plant. I don't have another slide later, so I'm going to mention this now. Plants get viruses. We have COVID-19. It's been a pain in the butt, or head, or sinus, whatever you want to call. Plants get viruses too. And what's interesting is that viruses move through the vasculature of the plant. So that means they can move through xylem and phloem. And the very tip of the growing plants, so lateral or apical meristems, or even root tips, the meristem is where you have active cell division and differentiation, which means that the xylem and phloem have not yet developed. It's sort of an undifferentiated middle where the meristem is. And because the xylem and phloem haven't reached it, the virus can't get to the tip. So in order to make virus-free plant stock, we go in with a microscope and we carefully cut out the apical meristem of the tip, and then we put it into tissue culture and they recreate the plant. And that way it's virus-free. So that's how we make virus-free plants. Now, looking at the callus technique, plants produce callus whenever there's a wound. So if you take a cutting from a plant, the fact that you took scissors or a knife and you made an incision, the act of cutting the tissues triggers a response, a wound response. And that wound response forms a bump. It closes off the wound by making sort of a lump of undifferentiated growth. And in tissue culture, we're kind of counting on that, right? So we are tricking the tissues to form callus. And in the picture here, you're seeing a bunch of undifferentiated, what we call friable callus. That means it can kind of crumble apart. It's loosely organized. And typically, this is done in tissue culture by balancing your hormones so that you confuse the plant. It doesn't know whether it should be growing shoots or roots. It just knows it should be growing. And so you get this sort of lump. If we look at a little closer at, say, an African violet cutting, you can see that in the center of the cutting, there's a lump. And it's starting to organize itself into roots here, right? But before it roots, it lumps. If you look at the stem, it sort of flares out at the end. In the greenhouse, when you take a cutting, you can tell if your cuttings are rooting well by just sliding your fingers along the stem. And you should be able to feel that the end of the cutting is starting to swell, and that means it's developing callus. So if we take tissue and we put it into tissue culture and we force it to form callus, then we can reproduce lots of plants by chopping that callus up into lots of little pieces and spreading it on the agar. And then we can change the hormones to get it to grow shoots and then transplant them onto another media that gets them to grow roots. And that's how we make lots and lots of plants. Looking at tissue culture, the X-plant, I should mention, is also the part that we're using. So I mentioned meristems, for example. We can take leaf punch discs. So if you take a hole punch and just snap a disc out of a leaf, basically any little piece of plant that you can put into tissue culture is an X-plant. This picture is just showing different things. We can use roots, stems, lateral meristems, leaf blade. This picture that I scanned really shows well how you can use a leaf punch to make callus. We can take a meristem to grow meristem, or we can take an embryo out of a seed. Cell culture is kind of interesting. That's where you take a sample of a plant and then you digest all the cell walls away with an enzyme. You end up with these little floaty balls of individual cells floating in suspension, and then you plate them onto your media. We call them protoplasts. So you can make a lot of plants in a hurry, but it's pretty extreme. So getting genetic consistency can be a challenge. Looking at the growing mediums for tissue culture, it all starts with a gel, typically a base. It's a lot like Jello. It's natural for the most part. And then we have all our typical fertilizer components: nitrogen, phosphorus, potassium, calcium, all the way down, micronutrients, macronutrients. Then there are vitamins. Now, we add vitamins because typically vitamins are important for organisms that can't make their own. Now, plants are where we get our vitamins from. But because it's an immature tissue and you want to make sure it succeeds, we supply vitamins externally just to make sure they're okay. Sugar is required as energy. This is an interesting one because this is the one time you'll see that I agree that sugars are fine for adding to plants in tissue culture because it's sterile. You get a lot of people who like to add molasses to their soil, but what they don't realize is the soil is not sterile. So by adding molasses, which is mostly sugar and salt with a bunch of other interesting, useful nutrients, the sugars feed the bacteria and yeast in the ground in the soil, and generally not the right ones. And you end up with a low oxygen situation, and the plant doesn't really do that well, or it's not helped as much as you might think. And then, of course, lastly, plant hormones are applied. I've got this picture because a lot of the vitamins that we use in tissue culture are also in your cereal. So when you're eating your breakfast, I always flip the box around, read the ingredients, look at the niacin and riboflavin, vitamin B1, all that stuff. Plants need it too. This slide, I don't want to spend a lot of time on it because I think you'll get this in other courses, but just really interesting slide showing you what the plant does with the different nutrients that we put into the media or into fertilizer for that matter. So take your time sometime, go back to that slide, look at it. It could be of interest to you. So as far as the hormones are concerned in tissue culture, we already know from the first part of the lesson that auxins stimulate cell elongation and root initiation, right? So we use auxins to initiate roots in tissue culture. And in plants, auxins travel from the top down, from the shoots down to the roots. So if you can remember that auxins travel down to the roots, they stimulate root growth, where cytokinins, which also stimulate growth, but in this case cell division, they travel from the roots up into the shoots to the buds. And so in tissue culture, we use cytokinin to stimulate the shoot growth or morphogenesis essentially, right? So balancing those two nutrients, getting the auxin and the cytokinin ratio together is important. Like I said earlier, if you get the balance just, where it's neither roots nor shoots, we stimulate callus development, which is undifferentiated growth. So tissue culture often is talked about in stages, so 0 through 4. If you're at a farm that's ordering tissue culture, you may order in tissue culture at stages 3 or 4. 4 is the best, obviously, because it's ready to transplant fairly quickly. Stage 3 is fairly immature. It's cheaper, but way harder to work with, so you want to avoid that if you have a chance. Let's look at a case study. So again, on the PowerPoint, you can look at more of the slides. I'm sort of skipping a few just to look at the key ones here. But this study is looking at both sugar and plant hormones on the impact of growing African violet leaf samples, sort of like what you tried in your lab. So here's a normal, balanced, we're getting shoots and roots all over on the cutting. It looks healthy. So that's when everything's going well. This is what happens when you forget to add sugar, right? So sugar is the energy. Sugar is the driving force behind laying down new cell walls and stuff like that. If you don't have energy, you can't go. It's like having no gas in your car. So there's very little growth. What growth you see is probably from what sugar was in that little piece of tissue when it was first cut, but not a lot. If you add a lot of sugar and all the other hormones are there, you get perfectly normal growth. Now, if you have cytokinin at ample supply but no auxin, remember cytokinin starts in the roots and goes up to the shoots, and auxin starts at the shoots and goes down to the roots. So cytokinin drives lots of shoot growth. So in this cutting, you can see there's all kinds of shoots forming, but we don't see roots coming out the sides. If we flip it, and we have lots of auxin but no cytokinin, there's roots coming all over the place, even on top of the cutting, growing up. It's just bonkers. So typically for successful tissue culture, we're balancing the sugars, the hormones, all the nutrients, and typically we have to customize that for every plant, although there's some common themes. And there's a lot of proprietary information around every particular variety that's grown out there. Anyways, that's a really a long, intense ride on plant hormones and plant tissue culture. I'm sure you'll learn more in your labs. Okay. Take care.